Welcome to Science Pills. So for this deep dive, we are generating a really comprehensive overview, a very serious but incredibly interesting conversation about a medical revolution that honestly sounds like pure science fiction.
Right. It sounds like something ripped straight out of a futuristic novel. But we are looking at the very real, very staggering frontier of 3D bioprinting and organ transplantation.
Yeah, and we've gathered all the latest discoveries, the clinical data, the foundational biology to really unpack this, because I want you to imagine waking up in a hospital bed.
It's a terrifying scenario.
It is. The lights are glaring, monitors are beeping, and a doctor walks in to tell you that your liver is failing. In our current reality, this is where the agonizing wait begins.
Exactly. You're handed a pager and put on a national transplant list with thousands of other desperate people.
You're essentially waiting for a tragedy to strike someone else so you might live. But what if, and this is the core of our deep dive today, what if the doctor just swabs the inside of your cheek, drops it in a vial, and says, give us a few weeks, we'll print you a new one?
It completely redefines how we conceptualize healing. And to understand why this is so critical, we really have to look at the sheer scale of the problem.
Because the current system is broken, right?
Fundamentally broken, yes. The global demand for healthy organs exponentially exceeds the available supply. Right now, only about 10% of patients who desperately need an organ actually receive one.
Wait, only 10%. So 9 out of 10 people just wait.
They wait. They are trapped in this medical purgatory, tethered to dialysis machines or artificial pumps, just running out of time as their biological systems slowly shut down.
That's incredibly sobering. And even for that lucky 10% who get the call and have a successful surgery, the battle isn't actually over, is it?
Far from it, actually. Receiving a traditional transplant comes with a massive, lifelong caveat. I mean, your immune system has evolved over millions of years to be this highly aggressive defense mechanism.
Right. Its whole job is to destroy foreign biological material.
Yeah. Precisely. So when you place someone else's liver or heart into your chest, your immune system immediately flags it as an invader. To stop your body from literally destroying the new organ, you have to take powerful immunosuppressive drugs.
Every single day. For the rest of your life.
Every single day. You are severely dampening your T cells, which creates a continuous physiological burden. You become incredibly vulnerable to opportunistic infections.
So a common cold could be life-threatening.
Exactly. Plus, you face an elevated risk of developing various cancers because your immune system is also supposed to clear out mutated cells. And that's not even touching on the immense financial burden.
So we're just trading an acute crisis for a chronic vulnerability. But sometimes, the most radical shifts in medicine don't start in a sterile corporate boardroom.
They often start with a deeply personal crisis.
Right. Which brings us to the origin story of a company called United Therapeutics. Back in 1996, Dr. Martine Rothblatt was facing a parent's worst nightmare. Her 7-year-old daughter, Genesis, was diagnosed with a rare, fatal condition called pulmonary arterial hypertension.
And just to give some context, PAH is an absolutely devastating diagnosis. The tiny blood vessels in the lungs become progressively thicker and narrower.
Which restricts blood flow, right?
Yeah, which means the right ventricle of the heart has to pump exponentially harder just to push blood through. Eventually, the heart muscle simply gives out. In the mid-90s, it was essentially an untreatable disease.
But Dr. Rothblatt, she already had this brilliant background in satellite communications, and she just refused to accept that. She literally went to the library, educated herself on the molecular biology, and founded United Therapeutics with one mission:
to save her daughter's life.
And remarkably, she did. She spearheaded therapies that managed the disease. Today, Genesis is alive and actually works at the biotech company her mother built for her.
It's a profound testament to urgent scientific inquiry. But what makes them so relevant to our deep dive is that they realize drugs are just a holding pattern.
Right. The drugs don't reverse the structural organ damage.
Exactly. To truly cure end-stage organ failure, you have to completely rethink the organ. You need to be able to replace it on demand.
Okay, let's unpack this. We're talking about moving from plastic medical devices to biological manufacturing, tissue engineering, and regenerative medicine, or TERM. But you can't print a human heart using desktop 3D printer plastic.
No, definitely not. You need materials that are alive. This is the foundational challenge of the field. We call the material bioink.
So what exactly is bioink?
Well, it's a highly complex microenvironment. It consists of 3 distinct components working together: the living cells, the biomaterials acting as a scaffold, and the bioactive molecules.
Those are the chemical signals, right?
Yeah, the signals that tell the cells how to behave.
Let's look at the scaffolding first, because if I just squirt a syringe of liver cells onto a glass dish, they're not going to stand up and form a 3D liver. They'll just be a flat puddle.
Exactly. They need a house to live in. In the human body, cells are anchored to the extracellular matrix, or the ECM.
The ECM, what does that actually look like?
Think of it as a microscopic, incredibly intricate 3D spiderweb made of proteins like collagen and elastin. It gives tissue its shape, it makes a liver feel solid, or an artery feel elastic.
And it lets the cells communicate too, right?
Yes, it transfers physical and chemical signals. So to recreate this in a bioprinter, researchers formulate polymers. We generally divide these into natural polymers and synthetic polymers.
Okay, let's start with the natural ones. What are we harvesting from nature?
Things like collagen, alginate, fibrin, and silk. Collagen is the most abundant protein in our bodies. Alginate is fascinating. It's derived from the cell walls of brown algae, or seaweed.
Seaweed. That's wild. And the benefit here is that they're highly biocompatible, right?
Yes, they have profound biological activity. When you embed cells into natural collagen, the cells thrive. They recognize the chemical markers and bind to the scaffold through receptor proteins called integrins.
So the cells basically think, okay, this feels like home.
Exactly. They can pull on it, proliferate, and secrete enzymes to slowly dissolve that temporary printed scaffold as they build their own natural tissue.
That sounds perfect. So why are we using anything else? What is the downside to the seaweed and collagen?
Physics and consistency. Natural polymers usually lack mechanical strength. They're soft, like weak gelatin. If you try to print something load-bearing like bone or a thick heart wall, it just sags and collapses under its own weight.
So they are biologically friendly, but physically flimsy.
Yeah. And the second issue is batch-to-batch variability. Harvesting alginate from kelp means the physical properties can vary by 15 to 30% depending on the season or the specific plant.
Wow, up to 30%.
Right. In a basic research lab, fine. But in a clinical setting, manufacturing an organ for a human, that unpredictability is a catastrophic nightmare for quality control.
You can't have the FDA approving an organ where the structural integrity depends on what week the seaweed was picked.
Precisely. Regulatory bodies demand absolute consistency, which is why we turn to synthetic polymers.
Enter the lab-made materials. The sources mention things like PLGA, PEGDA, and JAMA. What do these do that the natural ones can't?
They are engineered from the ground up. Think of them like infinitely tunable microscopic Lego bricks. You can precisely dictate how rigid they are and exactly how many weeks it takes them to harmlessly degrade in the body.
So they give you that robust mechanical strength and strict reproducibility. But there has to be a trade-off. What's the catch?
They are biologically silent. They lack those natural integrin binding sites. If natural polymers are a lush forest, synthetics are a sterile concrete box.
The cells survive, but they don't really thrive or organize.
Exactly. And some, like PLGA, release acidic byproducts as they break down. That lactic and glycolic acid drops the localized pH level, which can trigger an inflammatory response in the host tissue.
Which is the absolute last thing you want when implanting a delicate new organ. So we're stuck. Natural is weak and unpredictable. Synthetic is strong, but biologically sterile and potentially irritating. We need a hybrid.
And the field has a brilliant hybrid solution. It's called decellularized extracellular matrix, or DECM.
Let's dive into the mechanics of this because it's fascinating. The sources talk about taking a natural organ, say a pig's heart, and hooking it up to a machine that flushes detergents through it.
Yes, specialized detergents and enzymes.
And over a few days, it literally washes away every single living pig cell, flushing out the DNA. You're left with this pale, translucent, ghost-looking structure. No living cells left at all.
That is a perfect description. We call them ghost organs. You're left with the pure extracellular matrix, the naked architectural scaffolding of the organ, retaining all those complex, tissue-specific biochemical cues.
But hold on, if we use a matrix from a pig, why doesn't the human immune system attack it? We just said the immune system is hyper-aggressive against foreign tissue.
Because the immune system primarily recognizes foreign cells based on surface protein markers and foreign DNA. By completely stripping away the cells and genetic material, you remove the targets.
Ah, I see.
The remaining protein scaffold, the collagen, is highly conserved across mammalian species. A pig's collagen is structurally very similar to ours, so our immune system largely ignores it.
That's incredible. So you take this ghost matrix, grind it into a powder, liquefy it, and mix it with the strong synthetic polymers.
Exactly. You get a hybrid bioink, mechanical stability from the synthetics, and the rich biochemical signaling from the natural matrix.
Okay, we got the scaffolding sorted, the physical house is built, but a house is useless without people inside. We need the cellular fuel. And the sources are very clear that the engine driving all of this is stem cells.
Stem cells are the master builders of biology. They have two unique properties. First, they self-renew indefinitely, a limitless supply. Second, they are pluripotent, meaning they can differentiate into almost any specialized cell type.
Beating heart cells, filtering kidney cells, brain neurons.
Exactly.
Now, years ago, the conversation was heavily focused on embryonic stem cells, or ESCs, but the research shows a massive pivot away from that.
A profound pivot driven by ethics and biology. Embryonic stem cells are a blank slate, but they present the exact same immunological hurdle. The tissue still has a foreign genetic code, so the patient's body would reject it.
Right. So the monumental game changer was induced pluripotent stem cells, or iPSCs.
Yes, Nobel Prize-winning technology.
This is where personalized medicine becomes real. You need a liver, so doctors just take a skin biopsy or draw some blood. They isolate your adult skin cells and genetically force them to turn back the biological clock.
The mechanism is astonishing. They introduced genetic transcription factors, the Yamanaka factors, into the adult cell. It literally reprograms the epigenetic markers, wiping the slate clean.
So your adult skin cell reverts back to a foundational stem cell state, and because they're derived from you, they carry your exact genetic code.
That is the crucial breakthrough. An organ printed from your iPSCs is genetically identical to you. Your immune system scans it, recognizes your DNA, and categorizes it as self.
Boom! The risk of immune rejection is bypassed. No more lifelong immunosuppressive drugs.
Completely eliminate.
It's brilliant. Every brick in the house is alive and possesses your exact DNA. But I have a serious question about safety. We are utilizing cells engineered to rapidly divide and multiply endlessly. Aren't we risking just printing a tumor and putting it into someone's chest?
That is a highly astute concern. Safety is the paramount bottleneck right now. The main risk is indeed tumorigenicity. If even a small fraction of the iPSCs fail to differentiate properly.
You say they don't turn into art cells.
Right. If they remain undifferentiated and keep dividing uncontrollably, they can form tumors called teratomas. Furthermore, the reprogramming process uses viruses to insert the Yamanaka factors, which can occasionally trigger oncogenes, or cancer-causing genes.
So how is the field mitigating this? You can't just cross your fingers and hope for the best?
They rely on incredibly strict multi-layered protocols. Advanced purification techniques like flow cytometry filter out any residual undifferentiated cells.
So they essentially sift out the dangerous ones.
Yes. And all manufacturing must adhere to good manufacturing practice, or GMP, protocols.
Right.
Every cell line has its entire genome sequenced to check for mutations. The burden of proof for clinical safety is extraordinarily high.
Okay, let's assume we have the perfect bioink. Hybrid DECM hydrogel, loaded with purified patient stem cells. The ink is ready in a tube, but we need the hardware. How do you force living, fragile cells through a 3D printer without destroying them?
That is the central biomechanical challenge. Biological cells are delicate, too much pressure, heat, or radiation, and their membranes rupture. Different printing methods offer different compromises between precision, speed, and cell survival.
Let's start with extrusion-based bioprinting. It's like a desktop 3D printer, right? A syringe uses mechanical force to push the ink out of a microscopic nozzle layer by layer.
Extrusion is the workhorse of the field right now. Its huge advantage is handling incredibly high viscosities. You can pack a massive number of cells into a thick, dense paste,
which you need for dense tissues like heart muscle or bone. But the physics sound brutal. It's like forcing a water balloon through a tiny, rigid straw. The friction, the sheer stress, must be immense.
It is. The fluid dynamics create immense friction against the cell walls. And the data shows this sheer stress physically tears membranes apart, killing 20 to 30% of the cells during the printing process itself.
Losing a quarter of your living material right out of the nozzle seems like a massive biological deficit when you're trying to build a perfectly efficient organ.
It's the major trade-off. You get structural control, but you sacrifice a significant portion of your workforce to mechanical trauma.
So what's another approach?
We can look at inkjet bioprinting. It operates like the standard inkjet printer on your desk. It uses tiny heating elements or piezoelectric crystals to create a sudden pulse of pressure.
And that ejects tiny, perfectly controlled individual droplets of bioink. The advantages are speed and resolution, right? Placing cells with 50 micrometer accuracy.
The resolution is phenomenal. But the physics of droplet formation require the bioink to be highly liquid. It needs very low viscosity.
And because it's so watery, the printed structures are incredibly weak. They can't hold a 3D shape.
Exactly. You could print thin 2D layers of skin cells, but you could never build a sturdy, load-bearing piece of cartilage. The liquid would just spread out into a puddle.
So extrusion gives us strength, but crushes the cells. Inkjet is gentle and precise, but too watery. We need something that avoids the nozzle friction, but allows for solid structures. Which brings us to laser-assisted bioprinting.
This completely removes the nozzle, solving the shear stress problem. You have a donor ribbon, a glass slide coated with a thin layer of metal, like goals. Below the metal is your bioink.
And the printer fires a microscopic laser pulse through the glass, hitting the metal.
Right. It flash heats the metal in a millisecond, creating a tiny vapor bubble in the bioink below it. As the bubble expands, it pushes a precise droplet of bioink off the ribbon onto the substrate.
And because there's no narrow pipe, there's virtually zero shear stress. The sources say cell viability routinely exceeds 95%.
Over 95%, yes. It's a massive leap forward. But it has its own specific challenges. To rapidly solidify the bioink once it lands, these systems often rely on chemical cross-linking initiated by ultraviolet, or UV light.
And biology does not like UV light.
Not at all. Exposing rapidly dividing stem cells to UV radiation introduces phototoxicity risks. It can cause structural damage to the DNA. So you avoid the nozzle, but you risk mutating the genetic code with the curing light.
It's a constant balancing act. But this brings us to a fundamental problem none of these methods alone can solve. Let's say we use extrusion to print a thick heart. The real enemy isn't the nozzle, it's gravity.
Gravity is the absolute nemesis of soft tissue biofabrication.
Because a heart is a hollow muscle made of soft gel. If you print it in open air, long before the 12-hour print is done, the heavy upper layers will crush the soft bottom layers. You get a collapsed puddle of heart goo.
You simply cannot build a tall, soft, hollow structure in open air.
So to solve this, researchers developed the Pfeffer's technique free-form reversible embedding of suspended hydrogels. The mechanics are genius. You literally submerge the entire printer nozzle deep inside a container filled with a specialized gelatinous support bath.
Yes, you print the bioink inside this vat of gel. The bath acts as a Bingham plastic. It behaves like a solid to support the printed ink, but right at the moving nozzle tip, the mechanical force makes the gel act like a fluid.
So the nozzle glides through effortlessly, and as soon as it moves on, the gel instantly heals and acts solid again.
Exactly. It completely neutralizes gravity. The bioink is suspended, allowing researchers to construct astoundingly complex, overhanging, hollow structures like the interior chambers and thin valves of a human heart.
Structures that would be completely impossible in open air. And then once it's fully printed and chemically cured into a solid, how do you get it out of the gel block?
You simply raise the temperature to 37 degrees Celsius body temperature. The gelatin support bath gently melts away into a harmless liquid, and you pull out a perfectly intact 3D organ.
So what does this all mean? We have the bioinks, the patient stem cells, the hardware, and the FREsh support baths to neutralize gravity. It feels like we have all the puzzle pieces, but we don't.
We don't. Because if you pull a massive solid chunk of printed liver tissue out of that bath, you hit an impenetrable biological wall. The single greatest bottleneck in the entire field, vascularization.
Right. Printing a structure is only half the battle. If you print a solid block of dense liver tissue, the cells trapped dead in the center are going to suffocate and die.
It's a fundamental issue of biological scale. In the human body, almost every single cell is within about 100 to 200 micrometers of a capillary, about the width of a human hair.
Blood vessels are the supply lines. They deliver oxygen and take away toxic waste. But our printed block doesn't have any blood vessels.
Exactly. And printing takes hours. Oxygen can only diffuse through solid tissue for about 200 micrometers, so that the cells deep inside experience acute hypoxia-oxygen starvation. Within hours, the entire center of your printed organ undergoes necrosis. It literally rots from the inside out.
You have to print the plumbing. You have to print thousands of miles of microscopic hollow tunnels simultaneously inside the tissue. But how do you print a hollow tunnel inside soft gel without the surrounding weight instantly crushing it shut?
This is where we see the true genius of advanced material science. Specifically, the use of sacrificial inks.
This concept is so clever. The sources highlight a synthetic polymer called pluronic F127. It acts in reverse to normal physics. Water freezes when it's cold and melts when it's warm. This polymer gels and becomes a rigid solid at 4 degrees Celsius and turns into a flowing liquid at room temperature.
Yes, its inverse thermoreversible properties make it the ultimate sacrificial material. The printer lays down a highly complex 3D branching network using this cold solid gel. Think of a 3D map of a river system.
Then a second nozzle prints the actual organ tissue, the heart or liver biowink, completely burying this solid gel network. And because the gel is cold and hard, the soft tissue can't collapse into the space.
Precisely. It acts as a physical placeholder. Once the organ is fully printed and cured into a solid state, you take it out of the cold environment and bring it to room temperature. The solid pluronic F127 instantly melts into a liquid. You simply flush it out with a syringe,
Leaving a perfectly patterned network of hollow, interconnected, empty vessels running right through the center. But I have to push back here. Are these tiny hollow tunnels actually functioning like blood vessels, or are they literally just empty pipes? Because blood will immediately clot the second it touches raw polymer.
That is a critical distinction. An empty tunnel in a hydrogel is just a void. If you pumped human blood through it, the platelets would trigger the coagulation cascade and clot the entire system solid within minutes.
So how do you make it a real blood vessel?
It must be lined with a continuous layer of endothelial cells, the specialized cells that line your natural vascular system. They actively prevent clotting and regulate what molecules pass into the tissue. After flushing the sacrificial ink, researchers pump a liquid suspension of living endothelial cells through the channels.
And the cells just stick to the inner walls.
They do, heavily assisted by microfluidics. Microfluidic pumps initiate a continuous pulsatile flow of nutrient-rich fluid through the channels. The physical friction of the fluid creates localized shear stress.
Wait, I thought shear stress kills cells.
In a standard nozzle, yes. But here, a highly controlled, gentle shear stress is precisely what the endothelial cells need. It triggers mechanotransduction pathways, essentially teaching them how to behave. It forces them to flatten out, align with the flow, and form tight junctions. They organize into a fully functional biological barrier.
The physical act of flowing liquid tricks the cells into building a real blood vessel wall.
Exactly. And studies show vascularized cardiac patches have a 30% higher viability rate than non-vascularized ones. It's the difference between a thriving tissue and a necrotic mass.
Here's where it gets really interesting. Moving from theory to practice, let's look at the breathtaking progress happening across specific organs right now. Let's return to the lungs.
Looking at United Therapeutics, their progress is a masterclass in bioengineering. The human lung is arguably the most complex physical structure in the body, designed to maximize surface area for gas exchange.
The computational scale is hard to comprehend. To digitally map and print a lung scaffold, they use 44 trillion voxels. 44 trillion 3D pixels.
With that unprecedented resolution, they are attempting to recreate 200 million individual alveoli, the microscopic air sacs, intertwined with 2,500 miles of capillaries, all printed with the precision of a red blood cell.
And their stated goal is to advance these into human clinical trials in under 5 years. Under 5 years, it would completely upend pulmonary medicine. What about kidneys? Millions suffer from end-stage renal disease relying on brutal hemodialysis.
The sources highlight Trestle Biotherapeutics. They're using WITS Institute technology, deeply integrating developmental stem cell biology with 3D biofabrication. A kidney isn't just a sponge. It has highly specific filtering units called nephrons.
Right, the glomerulus that filters blood and the tubular system that reabsorbs water and makes urine.
Trestle isn't just printing a bean shape. They are attempting to print and mature specific functional nephrons, combining distinct stem cell lineages in precise geometries to coax the tissue into executing complex chemical filtration.
Let's shift to the heart. A heart doesn't just hold a shape, it needs to mechanically pump. Millions of muscle cells must receive an electrical signal and contract at the exact same millisecond.
Cardiac tissue engineering is unique because it relies on electrophysiological synchronization. Researchers can easily print cardiomyocytes, heart muscle cells, and they'll individually pulse in a dish. But getting the electrical signal to propagate rapidly across the whole patch is the major hurdle. Hydrogels are electrical insulators. They block the signal.
So how do you make squishy polymer gel conduct electricity? The research here astounded me. They're putting graphene into the bioink.
A brilliant application of materials science. Graphene is a single layer of carbon atoms with extraordinarily high electrical conductivity. By doping the bioink with graphene nanoparticles, researchers bridge the electrical gap between the cells.
It's like microscopic jumper cables embedded in the gel.
That's a highly accurate analogy. Data shows graphene-doped hydrogels improve electrical conductivity by an astonishing 300%. It allows the electrical firing signals to travel seamlessly, so the isolated heart tissue beats synchronously and powerfully, mimicking a living chest cavity.
Incredible. Let's move to the outside of the body, the skin. Severe burns are traditionally treated with skin grafts, causing secondary wounds, but bioprinting uses a radically different approach called in situ printing.
In situ simply means in place. Instead of printing tissue in a lab and transferring it, researchers use multi-axis robotic arms attached to extruders in the operating room. They literally print living skin cells, layer by precise layer, directly onto the patient's exposed burn wound.
And this works better than a graft.
Significantly. Preclinical data shows an 85% higher rate of successful vascular engraftment. The robotic system uses advanced optical sensors like LiDAR to map the exact 3D topography of the specific wound.
So it deposits fibroblasts in the lower layers for collagen and keratinocytes on top for the waterproof barrier, perfectly matching the wound bed.
Yes, drastically accelerating natural healing and reducing severe scarring.
Finally, we have to look at the brain. Printing neural tissue sounds impossible.
We are very far from printing a functioning human brain with consciousness. The architectural complexity is currently beyond our capabilities. However, researchers are making vital strides in localized therapies and disease modeling, printing neural support cells like astrocytes onto precisely patterned substrates.
The physical micropatterns actually act as a map guiding the growth of axons. But the most immediate application seems to be oncology, printing glioma stem cells into 3D tumor models.
Yes. A flat 2D petri dish fails to represent a tumor's physical reality. The outer cancer cells shield the inner hypoxic core from drugs. By bioprinting a highly realistic 3D model of a patient's specific brain tumor, oncologists can accurately test experimental chemotherapy drugs.
You can see exactly how the drug penetrates the mass in a lab setting before trying it on the patient's actual brain. Which transitions perfectly to our next topic. Before a surgeon sutures a 44 trillion voxel bioprinted lung into a living human being, how do they guarantee it will work?
We are entering what you could call the digital sandbox. This is where advanced bioengineering tightly intertwines with computer science, primarily through digital twins.
What exactly is a digital twin in this context?
It's a computationally massive virtual replica of a specific patient's physiology inside a supercomputer. Doctors input anatomical data from MRI scans, genetic sequencing, and real-time parameters like blood pressure. They simulate the patient. Then they implant the virtual organ into the virtual patient.
It's the biomedical equivalent of running flight simulations on a new airplane design. You force the digital plane through hurricanes to see where the wings snap before you put a pilot in it.
Excellent comparison. Using computational fluid dynamics, they simulate the precise hemodynamic flow of blood through the organ's vascular network. They can mathematically predict if blood pressure will cause a localized aneurysm, or test tissue compatibility to forecast immune rejection risks.
And artificial intelligence is doing the heavy lifting. Human engineers can't manually optimize the fractal branching patterns of millions of capillaries. AI neural networks generate those geometries. Machine learning is also revolutionizing the bioinks themselves.
Yes. Formulating bioink means balancing polymer concentration, cross-linking time, cellular density, temperature. Changing one variable affects everything. Traditionally, this took months of lab work. Now, AI simulates millions of combinations to predict the optimal recipe. It has reduced bioink development time by 40%.
But eventually, you have to leave the computer, you have to test physical tissue, enter organ-on-a-chip technology.
Also known as microphysiological systems. These are miniaturized, bioprinted fragments of human organs, like a tiny sliver of liver or heart tissue housed inside a transparent microchip.
Why use a tiny chip instead of testing on a lab mouse?
Because animal models are notoriously inaccurate. A mouse liver has entirely different metabolic pathways than a human liver. A drug that cures a mouse might be highly toxic to a human. With an organ on a chip utilizing human cells, you pump experimental drugs directly through the microchannels.
And microsensors perform real-time assessments of human tissue metabolism and toxicity.
Exactly. It powerfully bridges the gap between inaccurate animal testing and risky human clinical trials.
So if the technology is this advanced AI optimization, digital twins, 44 trillion voxel printers, sacrificial plumbing, why aren't bioprinted organs in every hospital right now? Why are 90% of people still dying on wait lists?
Because transitioning from the laboratory into widespread clinical use means facing immense roadblocks. The most significant is the biological bottleneck of functional tissue maturation. We've mastered the physical architecture, but making the cells perform their ultimate job remains elusive.
Wait, if we use the exact right stem cells and print the perfect shape, why doesn't it just work?
Because the cells don't immediately know how to do the heavy physiological lifting. An iPSC, differentiated into a liver hepatocyte in a lab, essentially possesses a fetal or neonatal phenotype. It's biologically immature.
So the printed liver looks like an adult liver on the outside, but structurally acts like a newborn infant's liver.
Precisely. The clinical data is humbling. A beautifully printed liver construct might only possess 20% of the metabolic enzyme activity of a real adult liver. A cardiac patch might pulse, but it lacks the powerful mechanical force to pump 5 liters of thick blood against systemic pressure.
The cellular workforce is there, but they haven't learned how to work together at full capacity. They need a biological workout in a bioreactor.
Yes, and developing bioreactors capable of providing the exact electrical, mechanical, and chemical stimulation needed to mature a massive solid organ is an engineering challenge all on its own.
That brings up the secondary roadblock: scalability and cost. Right now, this feels like bespoke, highly customized artisanal science, multi-million-dollar machinery, PhD teams, expensive biomaterials.
The current raw development cost is astronomical. But economic models offer a very optimistic outlook. The field is focusing on highly automated, closed-loop biomanufacturing factories. Projections suggest that by 2045, automated systems could drive the price of a bioprinted organ below $50,000.
Below $50,000. That makes it vastly economically competitive with traditional transplants, especially when you eliminate the hundreds of thousands of dollars spent on lifelong immunosuppressants.
It shifts from an expensive laboratory novelty to a highly viable, scalable healthcare solution.
But even if we solve the maturation and automate the factories to lower costs, we still have to deal with regulators. The FDA, the EMA. Reading the reports, this technology exists in an absolute regulatory Wild West.
It is in profound regulatory limbo. There's no unified global framework for bioprinted organs because they blur the lines between existing legal categories.
Because the FDA regulates medical devices like titanium hips separately from biologics like cell therapies.
Right. So is a bioprinted kidney a medical device because a robotic machine manufactured its structure? Or is it a biologic cell therapy because it's entirely composed of living human stem cells?
It's both. And neither.
Exactly. And the standardization problem of natural polymers rears its head here too. If your natural DECM ink has a biological variance of 15 to 30%, how does a regulatory agency approve it? Enforcing standardized quality control metrics on living, evolving biology is a massive bureaucratic nightmare.
But history shows science eventually pushes through these barriers. And as we conquer these roadblocks, we face profound philosophical questions. What happens when we can print body parts that are physically and biologically better than the ones we were naturally born with?
We cross from regenerative medicine into human enhancement. But before we reach pure science fiction, the field is preparing for the next technological leap: 4D bioprinting.
4D printing. I know the 3 spatial dimensions is the 4th dimension time.
Yes. 4D bioprinting integrates highly advanced, time-responsive, smart materials into the bioink. These polymers are engineered to physically evolve, actuate, and change shape post-fabrication in response to environmental triggers.
Give me a mechanical example of that.
Material scientists are developing temperature-sensitive hydrogels. A printer can rapidly deposit these as a simple, flat 2D sheet. But the polymer is programmed with a shape memory effect. When exposed to the exact temperature of the human body, it dynamically self-folds, rolling itself up into a complex tubular structure like an artery.
The printed object continues to build itself after the printer is turned off.
Yes. This self-assembling capability has been shown to reduce the need for highly complex external bioreactors by an astonishing 60%. The organ matures and shapes itself as it interacts with the patient's body.
That dynamic material science is mind-blowing. But taking that concept of programmable biology further, what happens when a healthy person wants an upgrade? What if someone requests bioprinted bones heavily reinforced with carbon nanotube polymers, making them virtually unbreakable?
Or artificial lungs with novel internal geometries that vastly increase oxygen exchange efficiency, so you can sprint for hours. Or cardiac muscle using synthetic pathways that reduce lactic acid, creating a heart that never fatigues. This raises an important question for society. What are the ethical implications?
Right, because initially this won't be universally accessible. Will this create a literal biological divide between the rich and the poor, vastly exacerbating global healthcare disparities on a genetic level?
The disparity argument is highly valid, and there are also immediate safety concerns. Using decellularized pig hearts, even washed of cells, introduces a severe zoonotic risk. You risk transferring latent animal viruses directly into the human population. How do we navigate the moral implications of blending animal matrices with human stem cells?
Let's assume we solve the economics and the viruses. We eradicate organ failure. We eliminate waiting lists. By striving for a culture entirely free of disease and physical imperfection, do we risk stripping away a fundamental part of the human experience? Our frailty has defined our art and empathy for millennia.
It is the defining question of transhumanism. At what point does medicine stop healing the sick and start manufacturing a new iteration of the human species?
To summarize the massive loops we've covered, we started with the basic bioinks, balancing natural matrices with synthetic polymers. We explored iPSCs, turning back the biological clock.
We examine the mechanical engineering protecting fragile stem cells from sheer stress, the FRE-Shache technique suspending gravity, and overcoming vascularization through sacrificial inks and microfluidics.
We looked at the awe-inspiring progress United Therapeutics mapping 44 trillion voxels for lungs, graphene nanoparticles synchronizing heart cells, and we navigated the digital sandboxes of AI and digital twins, acknowledging the sobering maturation and regulatory bottlenecks.
But the ultimate why is what truly matters. The multidisciplinary effort is staggering, but the objective is a world with absolute zero people on an organ wait list, an end to lifelong immunosuppressants. That is the incredible future this promises.
As we close today's deep dive, I want to leave you with a philosophical paradox. The ship of Theseus. If you have a wooden ship and over the years you replace every single rotting plank until none of the original timber remains, is it still the same ship? Now, apply that to yourself. If, over your life, you replace your failing organs one by one with perfect, technologically enhanced bioprinted replacements, at what point do you stop being you? When biology becomes a manufactured, upgradable technology, what happens to human identity?